please. So, Peter, do you want to give a bit introduction to your candidates?
Oh, yeah, sure. Yeah. Hi, everyone. Thanks for coming. So Nisha is currently a master at MQ at Macquarie University, and she has completed her master's there. And now she's applying for the PhD scholarship at UTS. She's original from Nepal. Stage is yours, Nisha.
Good afternoon, everyone. Thank you for having me. My name is Nisha Taman, and I am MSc PhD major biology. A little bit about my academic background. I have completed my bachelor's in environmental management with biology major from Tribhuvan University in 2016 from my home country, which is Nepal, which is in South Asia. That's where I belong from. And I have completed my master's in conservation biology from Macquarie University in 2020. I have mentioned some scholarships that I got throughout my university years, just showing off a little bit, I guess. So I will talk a little bit about my previous research experiences. Throughout my university years, I got amazing opportunities to be a part of a wide range of research programs. But I think these two projects that I worked on my final semester is something that has skilled me, that has provided me with skills to move forward with my PhD journey, and has given me the skill set that I'm confident that I can actually go forward and get my PhD. So I'll briefly go through this both project in my next slides. The first one is the phylogenetical and evolutionary analysis of Australian wild cotton species, Gossypium robinsoni. This is a very new cotton species, and I will start with why this project was very significant. Because we were trying to generate a rough or even a high quality genome for wild cotton species, because cotton is a very important and valuable economic crop. It has a very far-fetched economic implication, more than 150 countries being invested in import and export of cotton. It was, we're just trying to interrogate the genes of interest so we can create new cotton species with a range of phenotypical features such as high stress tolerance, insect repellents, and heat tolerance, especially if it's in Australia. So the methods we used were from the secondary source, we gathered more than 1 million scaffolds of long and short genome sequences, and we filtered them. So we got rid of genome sequences that was less than 400 base pairs, and we created FASTA files and we uploaded it in the Augustus for gene prediction. And we used various bioinformatic tools such as NCBI, CottonGen for genome sequence collection. So we can create a phylogenetic tree to track the evolution history. And we chose ITS, which is internal transcript spacer, to conduct the phylogenetic analysis. So you might be wondering why ITS, because it is believed to be conserved throughout the evolutionary history. And we created a phylogenetic tree for 34 diploid Gossypium species, and we checked the relationship and how far they are from each other to create hybrids. So our result was the closely associated species have similar chromosome morphology. So it's easier for us to create hybrids because it has normal meiotic combination and excessive F1 fertility. However, on the other hand, the hybridization among more distant relatives are frequently very challenging and unmanageable. And even if we manage to create hybrid with distant relatives, it has got basically very much of meiotic deformities. So that was my first project. The second project is literature review. It's movement ecology and dispersal ability and mating system of megalomorph spiders. This is a bit very different from what I have studied or what I usually study, but I was a part of this project because it's a literature review. And as a scientist, I think it's very important for us to study the papers to understand how meta-analysis work. And the aim of this study was to understand the factors that affects the movement of megalomorph spiders, and also what's the capacity of its dispersal, how far it is capable to disperse, and the mating techniques, like what sort of techniques it used to find mates. And the aim of this project was to find the papers, analyze the data, and conduct a meta-analysis to find the results. So the methods we used were, we used a website called Web of Knowledge for the literature search. We searched the literature, we arranged these specific keywords, and we extracted the papers. And we screened through them. We created an Excel data sheet with the columns such as species, the place where it's found, or the mating techniques and dispersal methods. And we extracted all the data from the papers that we collected, and we put it down on the Excel sheet for the results. So our result was pretty much a very significant results we got was megalomorphs are very small and restricted in distribution. However, it is found worldwide, which is very amazing because given that they are very restricted in the distribution capacity, they can be found everywhere in the world. And the project that I actually wanted to work on was a fine scale population genetic integration, given that they are so restricted in distribution, their genetical pool is very restricted. So that would be, I think, our second paper that I might get to work on future. Moving forward is the technical skills that I possess. So I think as a scientist, it's very important. Meta-analysis is very important to understand, to be able to read the papers from the past and to understand what works and what does not work within the scientific community. It will save us a lot of resources and lots of economic implications, I guess. And I also learned to operate various bioinformatic tools. Augustus is just one of the examples. And report writing is very important, as in when you move forward with paper publication and everything, report writing, experimental designing, research planning it's something very crucial. And the DNA extraction and PCR amplification, this is more of, I would say, something that I learned to work with in the lab, or more about safety, how you can use a lab properly, safety in the lab. These things are more implication on that. So my experimental skills is I have a very good understanding of genes and genome sequencing. Throughout my university years, I got to work very much with genes, phylogenetics, phylogeny of plants and animals. And I got to work with biotechnical tools, which helps in gene prediction mechanisms, phylogenetic analysis, DNA extractions, and PCR amplification, comparative genomics, metadata collection, and scientific literature review. Like I said, as a scientist, I think these are some of the very important skills that we require, especially going to paper publications and in the future. So this is the main project that I'm working on. This is my proposed PhD project, deciphering mitochondria morphology, organelle interaction, and mtDNA replication. So a little bit of background and context about the project. So as we all know, mitochondria is the powerhouse of the cell. That's a very basic knowledge. But what we might not know is every mitochondrion, which is a single mitochondria, contains multiple copies of mitochondrial DNA, which is mtDNA. And the replication, its separation, and circulation within the system is very important for maintenance and functions of mitochondria. And any mutation in mtDNA can contribute to mitochondrial dysfunction and can result in several human diseases, from cardiovascular diseases, neurodegenerative diseases, to different forms of cancer. And you can see that it is the second most diagnosed serious genetical disease after cystic fibrosis. This is the seriousness of the mitochondrial disease. So let's put this into data. It is estimated that 120,000 Australians carry the genetical mutation that can lead to mitochondrial diseases, with about 1 in 5,000 at risk of developing serious illness. And when we look at this data, we say, okay, 1 in 5,000, which is 0.0002%, and might not look very significant. But we have to understand that this is a simple, small population sample size. When you put this into bigger population sample size, the number goes up. And as a biomedical professional, this number is not just data, it's an individual. And we're looking at the social impact, personal impact, economic impact that disease is having on an individual. And recently in 2021, December of 2021, the legislation has been passed, which is known as May's Law. And this has legalized the partial DNA donation, allowing women to give birth without passing on a genetical disease. And putting this into perspective, how this works, what they do is they scoop out the affected empty DNA out of the egg, and they implant the donated empty DNA out of the donor mother's egg. And it is amazing because at the end of the day, this new baby has genetical information from 3 different people. And this was illegal in Australia before to perform any experiment on stem cells, but now it is legalized. So this is a big leap of faith towards science from Australian government. And now let's talk a little bit about mitocytosis. Mitocytosis is a process in which cell gets rid of the infected mitochondria. You'll be hearing this word a lot in upcoming slides. I just wanted to introduce you a little bit about this word. So moving on to our hypothesis. So our hypothesis is that TFAM, which is also known as mitochondrial transcription factor A protein, is very crucial for the maintenance of empty DNA and cell survival. And the function of TFAM is determining the abundance of mitochondria genome by regulating the packing, stability, and replication. So let's talk a little bit about organelle interactions. The crosstalk between mitochondria and other organelles is very important in tumorigenesis and formation of tumor. And the organelles work as an entity. They can't work by themselves. They work as a community. So any impairment in the relevant cascade may lead to the change in cell microenvironment within the cell and may lead into activation of certain oncogenes and mitochondrial genome mutation, which then leads into cancer. So now we're going to talk about mitocytosis. Our hypothesis is that mitocytosis helps in getting rid of the empty DNA mutations by selectively disposing the mutated empty DNAs. And we believe that by continually removing these damaged mitochondria, mitocytosis can help cells avoid this adverse effect of accumulating of dysfunctional mitochondria. And we can imagine what happens if we have dysfunctional mitochondria accumulating within the cell. It kills the cell. And we know if it kills us, if our cells start to die, what happens to us? So we have a hypothesis that if we can induce mitocytosis, it might help us to curb mitochondrial diseases. Moving forward to what our aims are from our project is that we want to apply advanced imaging to visualize the empty DNA structures, to understand how its replication and transcription works, also what's its correlation between the morphology and how it interacts with other organelles. And we want to systematically pinpoint the molecular mechanisms between empty DNA replication and transcription and its correlation with morphology and its interaction within the organelles. So our other aims is to analyze the role of mitocytosis, we have already talked about, in mutant mitochondrial disposal, to see in what level does it help to get rid of the mitochondria that has been infected, and to identify the functional genes that's responsible for mitocytosis, to see what genes actually are required to induce mitocytosis. And we also want to study the role of mitocytosis in identifying mitochondrial diseases in its early phase. So far, it's really hard to identify mitochondrial diseases when it's in its start phase. It's usually it's only figured out when it's in its final stage, or it is really, it is to a point where it cannot be further treated anymore. But we're just trying to see if a cell is producing high amount of mitocytosis, if the reason is because they have got some infected mitochondria and they're trying to get rid of it. So what are the methodologies that we're using to do this? We are going to use the cell cultures. We're going to study the molecular biology of what's happening within the cell, within the mitochondria, and how the replication and transcription is happening within the cell. We're going to label the live cells. We're going to use the imaging technique, and we're going to track the single molecules. We're going to use the immunostaining to protect which helps us to preserve the cells. And we're going to use a super resolution imaging to see the changes within the cell. And the last part, which is gene editing and clinical animal model trials, we are going to do this in Professor Phil Hansborough's lab, because our lab is technically not allowed to trial animal models. And when we talk about gene editing, we're talking about inducing mitocytosis within the cell. And what's the significance of the project? The significance is that the direct visualization method and the better understanding of mtDNA structure will help us to decipher how disease work within mitochondria and how human-related. So we can be able to understand a lot of information on how it works within a molecular mechanism, mitochondrial dynamics, and mitochondrial information network formation help us to decipher how human diseases work and how it functions within the cell system. And this project will also play a significant role in future for advancement of therapy related to mitochondrial dysfunctions and morphological abnormalities to numerous life-threatening diseases in which, as we can all see, that mitochondrial dysfunction plays a very vital role. So what are our limitations? So our limitations are that there is not enough studies on organelle interaction. So it's very hard for us to understand how mitochondria reacts with different organelles and how it helps in disease spreading. And the cancer has a huge heterogeneity. So it's hard for us to understand where the cancer really starts from. Maybe it's a cell, tissue, organ, it could be anything. So it is very important to investigate these things using animal models, which will be more substantial for clinical application and drug development in the future. And it has been suggested recently that a mitochondrial nucleoid, not the mtDNA, is the unit of mitochondrial genome inheritance. And knockdown of important protein like TFAM is not only results in unusual nucleoid, but also causes asymmetric transmission of mtDNA into two daughter cells. And so thus we need very significant interest in its structures and its regulation to address the underlying mechanism of mitochondrial diseases. And TFAM, which is said to be very essential in mtDNA maintenance, has so far never been linked to a human pathology. So it's something that needs to be researched on. And according to a paper from JIO 2021, they say that there are like specific protein coding genes required for mitocytosis. However, we need enough research to support this conclusion. So this is a timeline of what we are trying to go in 3 years of our research time and what are the aims that we're trying to meet within each year of the research. So the first year will be more focused on training, study of molecular biology, getting the cell cultures and analyzing the data. The second year will be more hectic and we'll be doing more on labeling the cell, imaging, tracking the single molecules and staining, using the immunostaining with the fixed cells and super resolution microscopy to gather the data so we can use it for the third year, which is more of animal gene editing and clinical animal model trial, which will be happening in Professor Phil Hansborough's lab, who is the director of centenary of UTS Center for Inflammation. And we'll be working on, in the last year, we'll be working on analyzing all the data we have collected throughout first and second year, and we'll be working on paper publication. So moving forward to why UTS. UTS is the youngest, one of the youngest university, and it has made to a top 150 in world universities ranking, which is amazing for a young university like UTS. With more than 150 industry partners, the students have got enough marketplace to use their academic skills And cross-discipline research opportunities that UTS provides is really amazing, because I think cross-discipline research is something I'm always interested in. And it also helps to challenge, it also helps to address the challenges of future and present. And as an international student, the student services and support is very important for me. It has always been, because international students have to deal with a lot. And UTS has a wide range of student support and services, which is really amazing. And also they provide with a range of scholarships. And why biomedical school? I think we're in an age where we don't see science as faculties. We don't see physics, chemistry, or biology apart from each other, but we see them as a complete body of science. And biomedical engineering is something that includes the adventures of everything, and works for a greater purpose of innovation and cutting-edge technology to improve human quality of life. And when I read this book, The Life on the Edge, The Age of Quantum Biology, it is absolutely amazing to see how far science have come in terms of technology, in terms of understanding universe, in terms of understanding quantum phenomena, but also it is very important to understand that we have so much more to figure out. And biomedical engineering is something that will help me to widen my understanding of science and will give me the freedom to explore every avenues of curiosity, I think, without restricting my knowledge within a faculty. So why Quanzu Lab? Why my supervisor? I think this is the easiest answer for me, and I'll absolutely say it's because of Dr. Sue. The love, support, and respect that Peter has for his student is incredible. To get this project ready, the time he has invested in, the support he has provided is absolutely phenomenal. And you can have the supervisor with the fanciest lab or the most amazing technologies, but if he or she does not support you, it's really hard for a student to go through that. And I think I don't have to worry about that with Dr. Sue. And I did email a couple of potential supervisors from within UTS. However, the project that Dr. Sue is working on is very valuable and far-fetching. And his nature of work actually aligns with my future objectives and my career goals. So there was no second thinking on that. I wanted to work with Dr. Sue, and I want to work with Dr. Sue. So my career goal for next 5 years, I would say I want to see myself as a research scientist and work in a research project that would contribute to the future of medicine and science and technology. And I have mentioned some potential collaborations that I might get opportunities to work with if I made it through this interview. And why am I the right candidate? Academically speaking, my background of research-focused master's degree and gaining a valuable experiences of using various bioinformatic tools and the study of my genes and evolutions can help a lot. And my contribution in literature review as a second author assisted me to learn a lot of data collection, meta-analysis, which helped me to garner a lot of meaningful skills to contribute to my PhD journey. And personally, I would say I have a very strong work ethic, and I believe in integrity, and I believe in excellence. And I'm very disciplined individual. I'm passionate, I'm a very curious person. And like Einstein said, never stop questioning. And any scientific ordeal always starts with a question. You question why, and then you figure out why. So I think being curious helps me every single day to be a scientist. And I'm confident that if I am given this opportunity, I'll thrive and deliver quality work within stipulated deadlines, because I understand deadlines are very important for organizations. And as a woman in engineering, I will support and motivate more women to be in science. And because I come from a different cultural background, and this is a completely different country. When I first started my university, I could not understand Australian accent. It was really hard for me. So I understand how important it is to support multicultural representation. And lastly, I would like to say thank you. I was just caught by surprise because I was thinking I'll start at 3 and then I'll see someone give their first presentation, and I'll just get lost and give it a bit of a try, and then I'll come back at 3 and I'll give my presentation. But oh well, I just got the opportunity and I said, why not? Thank you so much, everyone.
Thank you very much, Nisha. That's it is a great courage that you go first where you just arrange lastly. And so any like questions from the audience for Nisha?
Yes, I raise my hand if I can start.
Yeah.
Thank you. It was a great talk. I think you're clearly a good public speaker, and that was clear and logical. I have a few questions. Let's start with your previous work. So you said that you were involved in sequencing the cotton genome.
Yeah.
So which part of this work you've been involved? Collecting the DNA, sequencing the DNA, or analyzing the?
So
You know?
it was a big PhD project. It was someone else's PhD project. So I was just there doing my last year of university. So what I did was I actually worked with him first and to analyze I worked in the part of gene prediction, collecting the genome sequences of other cotton species to create the phylogenetic tree. I created the phylogenetic tree, assisted him to create the phylogenetic tree and to understand the evolution history.
What was the ploidity level of the cotton you guys work with?
The C genome.
What was the ploidity level of the genome?
I think it's sorry, that was my really, I did it 2 years ago. I almost forgot about it.
That's okay. So because I think if it's a multiploid genome, it might impose some problems with mapping with the multitude, genes, because the chromosome number is multiplied.
Yeah. Sorry, let me just have a look. I just.
No, I mean, no, you don't have to look. So yeah. The other question I have is related to your previous project. You mentioned that you're gonna use gene editing. What you're gonna edit and what you're gonna use with editing, and why you're gonna use gene editing? What's gonna be the genes or the
So we have got few protein coding genes. So KIF5B, DRP1, myosin L9. So this has already been Peter's co-supervisor has actually written this paper called Cell, and it has shown us a couple of protein coding genes that is responsible for mitocytosis. So when we say gene editing, we're saying we're trying to induce mitocytosis within the cell to see if it gets rid of the mutant mitochondria, to see if that helps in disease. It helps to curb the disease, mitochondrial diseases.
So what gene editing technology you're planning to use for inducing gene expression?
I would say we might see if the high amount of certain proteins so we're going to take some proteins that are really required for mitocytosis, that are required for inducing mitocytosis, and we're going to see if the high amount of those protein actually induces mitocytosis within the cell.
So you're gonna overexpress those proteins, or you're gonna induce?
Yes, we're going to overexpress the proteins and see if it helps in inducing mitocytosis within the cell.
Is that correct, Peter?
Partially? That's my fault.
No, I mean, I'm curious. I'm not judgmental here, just because the past few talks, people using gene editing technology for induction, and gene editing historically is knocking down gene expression. There are ways to do, but I wanted to know the technical details. Not right now at the moment, but in general. So
in my mind, I think CRISPR would be a candidate.
But in that imposes the question how you use CRISPR for gene induction, right? So that's a very tricky that's a very ingenious approach, but you have to know it if you. My last question is, how do you see yourself if you have already the PhD 5 or 10 years from the PhD? So what's your plan in the long term? You said.
when I chose biomedical science, the only thing that I had in my mind was that I want to contribute to science and technology and medicine, because I don't want myself restricted in a certain faculty. So I thought if I be a research scientist, I will get the chance to work in cross faculty research programs. So I have got this idea that if I become the scientist in next 5 years, which I want to become the scientist in next 5 years, I would work in various research project that is cross faculty. And if I work with Dr. Su, I have got some really good people to contribute with.
Thank you. No more questions from me.
Okay, I think because of the time limit, maybe we can go to the next candidates. I will send like the recording to other like the school members afterwards. I think maybe
I can suggest that you can send questions to Nisha in the chat, and she can answer.
Yeah, I can do that.
We can continue the review process behind the, right?
Yeah, sure. Hachi, do you want to do the next presentation?
Next time, these two students have problem with team.
Yeah, I know. Yeah, maybe because of the overseas. So we finish with our Zoom candidates first, and including Hachi and also Helen's one candidate. Okay, I'll give Hachi a bit introduction. So Hachi has already been working with me. Since his master project has been working with me for almost 1 year, and he's now still continuing working with me as a research assistant, and he helped a lot for me to establish our lab protocol for like exosome-based analysis. I think it's a good time, and he's ready to take it as a more systematic training through a PhD project. Okay, now Hachi is time is yours.
Okay. I'll share my screen. So can I start now? Yeah. Okay. Good afternoon, everyone. I'm very glad you can join my interview. My name is Haoqi Mei, and I'm making this presentation for my PhD application. In my presentation, I will briefly introduce myself first, and then I will talk about my previous experience and my professional skills and my previous research project. And also I will talk about some project I'm going to do during my PhD degree. At the end of this presentation, I'm glad you can ask me some questions. So I'll start from the first part, my self-introduction. I'm an international student. I'm come from China. I did both my bachelor and master degree at UTS, and my bachelor degree is the Bachelor of Biomedical Science, which is the laboratory medicine-based course. My core subjects include histology, immunology, anatomical pathology. During my master degree, I changed my major to the biomedical engineering because I found some medical diagnosis and treatment currently is still facing some unsolved problems, and the biomedical engineering is to develop the technologies to solve this problem. So I changed my major. During the study, I started touch some like precision oncology-based subjects like the bio-nanotechnology, which is the target drug delivery system. And I did my previous research project, Biomedical Engineering Project A, and I will talk about this project later. During the 2017, I did an internship subject in the microbiology department of Royal North Shore Hospital. And during this period, I performed a range of diagnosis tests, including the fast antigen test for the meningitis, the caused by Legionella, and the strep pneumo, and also urinary tract infection diagnosis and a PCR for the TBs. From 2020, I started doing the tutor for biomedical introduction of biomedical engineering. My responsibility was included the talk about with the students to help with them with their projects. And also I will answer the question and have other lecture activities. During the talk about my professional skills, I accumulated a lot of laboratory skills during my previous study and my internship in the hospital. For example, my previous hospital internship and my previous study makes me have sufficient technical skills and make me getting familiar with the clinical diagnostic method and workflow. Also, and I always cooperate with my classmates and colleagues during my previous study and interns. So I have a very strong team working skills. The tutor experience and my previous volunteers experience in UTS helps and makes me have a strong communication skills to talk with others. The reason why I choose to do the PhD, firstly, I think it is my parents' influence. I was born in a medical background family, and my father is a doctor, and my mother is a pharmacist. So when I was a child, I have many chances to visit their workplace and to see how they help the patients. And also, they always talk about their work experience at home, so I can hear that. So I become very interested in this area, and I was trying to become the medical research in the future. And talk about why I choose the UTS, because I've been studying UTS for 6 years. I'm familiar with everything in UTS. So, and I believe I can get very good high education in the UTS. So I choose to keep doing my PhD at UTS. My previous project during the master degree, I started to do the project from 2020. I was going to do some cancer research during that period. And I started to contact Dr. Ying because I found Ying is very active and emerging research in this area. And by talking with Ying, I started to understand the emerging technology for cancer diagnosis, which is the liquid biopsy, and very potential materials, extracellular vesicles. So I started to do my first research project, which is the investigation of EEV and as the biomarker for the pancreatic cancer early detection. So the pancreatic cancer is one of the most deadly cancer. The 5-year survival rate is very low, which is lower than 10%. Because at early stage, the most of patients with pancreatic cancer are asymptomatic, or they can only have very non-specific signs like the abdominal pain or other gastrointestinal disorders. The current diagnosis for the pancreatic cancer is mainly about the medical imaging screening like CT, MRI, and PET scan, combined with the histopathology diagnosis. But all these methods are unable to provide early detection because they need to based on the clinical suspension first. But this is very hard to find out if the patient have the pancreatic cancer. The liquid biopsy is the one emerging technology for the cancer diagnosis. This kind of diagnosis is to detect biomarkers in the body fluids, like the urine and peripheral blood. And the current biomarkers for liquid biopsy are focused on the circulating DNA and circulating tumor cells. They have a very good development in these two biomaterials, but they're still facing some challenge. So, for example, the circulating DNA is very short half-life in the circulation, so it's not very easy for the detection. And also, the circulating tumor cells is very rare in the circulation as well. So they facing some problems to in the actual test. And also both materials are mainly for the treatment monitoring, not for the early diagnosis. So now a new material, which is called the extracellular vesicles, and become a very potential biomarkers for liquid biopsy. These vesicles are released by all kinds of cells, and they have a very similar biological composition to their parental cells, and carry a range of biomaterials like the DNA, RNA, proteins, because they can released by both pathogens and normal cells, so they can reflect both physiological regulation and pathological things. This is the works I did before. I did the cell culture, and also I made a protocol for the EV isolation. I used a range of technology to validate EVs, including the cryo ETM and a nanoparticle track analysis. And I also I established the Western blot protocol for the EV membrane protein investigation. This is some result from my previous research, and we use the normal microscope to validate EVs, and we also use the cryo ETM to show their detailed structures, and we also use the nanoparticle track analysis to investigate the extracellular vesicles, the size range. So this is the Western blot, the result from my previous study. And we test the several EV markers and the cancer markers. And we found all the cancer cell released EVs have very high expression of the cancer markers, and which is matched to our cell result. And during my master degree, I also helped the Professor Jing's group from IBMD with their Western blot analysis. And the paper that I'm co-authored is currently in preparation, and that will be submitted soon. So here, this is my project I'm going to do during my PhD degree, which is a single vesicle, extracellular vesicle analysis for the pancreatic cancer liquid biopsy. Generally, the target is to develop a non-invasive invasive diagnosis for the pancreatic cancer detection, and to save the time, to improve the time limit and make very accuracy. And this project divided into 3 stages. And firstly, I will develop a bioassay for the AV analysis. And all these methods will suitable for clinical use. And also I will test the EV biomarkers from current research, and to detect whether they are possible to do the early detection for pancreatic cancer. And also all these tests will be investigated. The clinical performance of all of these methods will be investigated. So this is my PhD innovation. The firstly is the EV analysis, single EV analysis. So currently the EV analysis is based on the bulk EV analysis. And because even though these EVs can release from the same parental cells, they have a very, some subpopulations, and because they have different compensations. And if we use the bulk EV analysis, some heterogeneity will be masked and have a misinterpretation of actual target. So currently there was some single EV detection technologies, like this one. This is a digital ELISA, and also the digital PCR as well. But all these methods, they shows they use the low throughput detection, so they cannot detect many samples in the same time. So in my PhD degree, I'm going to use a flow cytometry as my EV analysis method. Firstly, the flow cytometry is a high throughput analytical instrument, and also they are routine used device in the clinic. And so but the conventional flow cytometry, they can only detect the large particles, which is around 500 nanometers. And so to solve these problems, I'm going to apply the aptamer to enlarge the EV signals for the conventional flow cytometry to detection. And I think if I can use the flow cytometry, use my EV detection, that would be helpful for the transfer my research outcome to the clinic. Here's the general workflow I'm going to do during my PhD degree. And so here's the single EV analysis. So there are two different lines. One is using cell line as the module. So I will do a cell culture first and do the EV isolation and use the aptamer based flow cytometry to do the analysis to find out if they can detect the EVs. And also, I will use the patient's plasma as the module as well. So the patient's plasma will be collected from the hospital, and the plasma-derived EV will be isolated. And I will use the flow cytometry, which I've established, to test these cancer markers in the patient's plasma. And finally, their performance, clinical performance will be investigated. And this is my research timeline. And the patient recruitment will cover the whole period of my study, which is 3 years. And at the beginning of 2022, I will start some literature review to have a deep understanding on this area and this technology I'm going to use. And then I will start to establish the bioassay, which is the flow cytometry detection. And start from 2023, I will focus on the EV biomarker investigation to find out their capability for the pancreatic cancer detection. And start from 2024, I will investigate the clinical performance of my research. And at the last half year, I will focus on my final thesis and the paper publication. This is my supervisor team. First one, Dr. Ying, is my proposed primary supervisor, will provide support during the whole period of PhD. And also we have two world-leading pancreatic cancer researcher, but the oncologist. First one is Professor Phoebe Phillips from UNSW. Another one is David Gostin from Principal Wells Hospital. They will provide the oncology support during my PhD degree. And also we have started to collaborate with Wenwang Zhong from University of California to develop the DNA amplification-based flow cytometry. I'm very glad you can join my interview. Thank you very much. And any questions?
Thank you, Haoqi. Any questions from the audience?
Yes, Haoqi, very good presentation. I have a couple of technical questions. One is a general one. What is the benefit of single EV detection in the clinical setting?
So that's the current, if we use the bulk analysis, so some like exact the bio content cannot be interpreted. So if we use a single analysis, so we'll see the very detailed of their composition differences. So we can find out the heterogeneity, which will very important for the further diagnosis and treatment.
No, that's not, in the clinical setting, is different. I mean, basic science is very different with the clinical setting. My question was clinical setting. What a clinician can benefit from a single EV detection or analysis? I mean, scientists curious to know what is what. That answer was related to the science, most probably. But in context of clinical settings, I cannot justify anything, any usage for myself for the single exosome or EV detection. Bulk or interpretation of change in the load presence or omission of them in certain ecosystem of the disease before prior to treatment, after treatment. That makes sense, but I just cannot find a logical explanation why a single EV is useful for a clinician. Even a single cell is not useful for a clinician. Do you agree with me or no?
Yeah, I got your question. I think this is probably a bit hard for how to understand, like answer this question. So I can like have some discussion with you. What I think is that obviously like a single cell analysis, single EV analysis is a trend in this field. Like you can get like high impact factor publications and that. And to be honest, it is true that the single EV analysis, there are lots of great work, but the clinical relevance has not been established yet. And what we can find that if we can do that and then get a more informative information from the single EV analysis, and that may brings to like a more accurate decision of the biomarker panels. Obviously, if we do like a single EV analysis, there will be a lot of data involved. We can also like implement like a machine learning to have a good like algorithm for the data analysis, and we can then introduce to the clinician too.
But I will put my bet. I will put my bets onto optimizing a robust flow cytometry protocol, as he said, that a clinician or a scientist can routinely use it. Imagine that a clinician always give you half ml of plasma, 1 ml of plasma, consistently want reproducible result of characterization of the EVs for certain biomarkers. There is no even a robust protocol like that that anybody could use in hospitals.
Yeah.
I would, if I was about to do a PhD, I would spend it like that, because at the end, while you can still publish good quality papers, you can assure that 3 years of your efforts can be utilized by many scientists and clinicians down the road. That's something that to me is missing in this field, building robust or reproducible protocols. Well, anyway, that was very good for a good undergraduate research, and I hope that you could continue this in your PhD.
Any other questions?
Yes,
Yeah,
I have.
can I ask you a question? I mean, it was not clear to me whether you want to develop a EV marker for the diagnostic purpose or for the response to therapy or what? I mean, it's a totally different game, between say, you identify someone who has high risk of pancreatic cancer to, something to do with diagnostics or respond to treatments. And I wasn't clear, what was the outcome of the proposed projects.
So the outcomes for my proposed research will be if I can develop these tests and use a routine used technology in clinic, it can directly, I think it can be directly used for clinic or the cancer. So we focus on some like the high risk patients. For example, they have the chronic pancreatic issue.
So you are talking about screening, diagnostics.
Yeah.
Right. And screening, it's not about clinical setting, okay? Screening is a different game. It's about epidemiologies. And that, lead me to the questions that because you focus your second year on the patient recruitment, but because it is a screening purpose, you also need a control group, not just a patient, right? I mean, imagine that, you have a EV test, with the sensitivity of, let's say, 80% if you are lucky enough, and specificity of, say, 90%, if you are very lucky enough to get that, then you still, need about 10-fold difference between the controls and the patients. And are you sure you're going to get that, within 1 year?
So, sorry?
So we Actually, there is a difference between screening and an early diagnosis. I just find this out myself. Actually,
Yeah,
early
you know,
diagnosis is to assess that patients
but
with
he's
some symptoms.
talking about
Yeah.
screening, and that's
No,
what
we're not we're not doing it at the screening at the moment. We're doing early diagnosis with symptomatic, like a patient with symptoms.
okay. So if you want to do an early
Yes.
diagnosis, then you still need to demonstrate that your EV marker
Mhm.
will have a certain degree of sensitivity and specificity. And doing so, you still need to have a substantial control groups,
Yeah,
meaning people
we
without
currently
cancer.
recruiting like participate from Red Cross blood life blood. So that's where we assess our healthy participant. We're now preparing a survey form to get their health information to have a more better like a control group. But I believe that's not like a 100% good. We can figure out this later once we collect more
Yeah,
like a preliminary data for the
I
control
guess
group.
you better focus on your technical accomplishment rather than clinical application, because to translate from some discovery or some new thing to clinical application, it takes, trust me, it takes more than 10 years.
Yeah, absolutely.
So, and you have only 3 years to go. All right. So, do not be too ambitious. Yeah.
Thank you, Tuan, for your suggestion.
Okay.
Yeah, I have one question
Mhm.
about so regarding your PhD and after the PhD, how do you do with your outcomes? Apart from the application, I know you will have the publications.
So after my PhD, I will keep I think that everything is based on the investigation of the clinical outcomes. If the outcomes get very positive result, I think I can suggest to firstly, I think we can enlarge the sample collection for the patient samples. And then if we continue to get very positive result, I probably can suggest to apply our technology for the clinical use.
How do you apply your technology for the clinical use? Do you have some idea, like how do you?
Wow, it's quite a tough question. Actually, so far I just considering is that everything is based on my PhD research, and
For example, if your outcome perfect, beautiful, everything's perfect, what do you do after this?
I think it's for the paper, the publication, and
It's hard. Yeah,
I think that's a question for me, not for us. I don't have.
Okay, that's fine. That's why you join the PhD. You will know how to.
The next step, if you got
First,
the
enjoy
perfect
your
technique,
PhD.
you are going to work with the Royal College of Physicians. Okay, so that's where you have to work with them to convince them that it can be applied to clinical practice. I can show you, some trick about that, but no, that's a long way.
I have a quick question. So I'm going to ask you the same question that for the previous candidate. So what's your future goal? What's your long-term goal if you've been awarded with a PhD?
Sorry, could you repeat your question? I didn't hear it clear.
So what's your long-term goal? How do you see yourself 5-10 years from the PhD awarded? So what's your long-term goal? If you
I think only 3 years.
No, but how you see yourself as a researcher, or how you utilize the PhD in 5 years after you graduated as a PhD student? I just want to know what your future goals. Why are you doing the PhD?
I just try to become a medical research in a cancer diagnosis area, and I think this because I said my previous study is on this area, and I just try to continually doing that because I'm very interested in here by doing this.
So you want to be an independent researcher with your own research group? That's your plan?
Academic or industry, things like that.
I think I just focus on the research, just not just keep doing the research work. It's not just academy, I think.
Okay, thank you.
Okay, I think it's time for our next candidate. Helen,
Yep.
you want to introduce your candidate?
Sure. Haji, would you like to, could you
sure.
just stop sharing your screen? Thank you. The next PhD applicant is Prakriti. Prakriti, you here? I think you are. Oh, sure. Yeah. Prakriti Svalkoti is the honor student. Have been working with her for a full year, right? A full year since last February to this February. Prakriti did a really great job for one part of our GDRF grant. So that's actually quite essential and basic, but she successfully finished that and then did a very good preliminary data collection. So and then after this, during this honors project, Prakriti expressed her interest to continue work on this project. That's why we apply for the PhD scholarship from UTS. So Prakriti, would you like to start?
Yes.
Go ahead.
Hi everyone, I am Prakriti and I am glad that I got an opportunity to attend this PhD interview. So I will be working with Dr. Helen Chu as a primary supervisor who is lecturer at School of Biomedical Engineering. My co-supervisor will be Dr. Lana MC Clements, she is a senior lecturer at School of Life Science. And my industrial co-supervisor will be Professor Bernard Took. He is director at Australian Foundation for Diabetes Research. So my academic background, I completed my Bachelor of Science in Microbiology from 2014 to 2018 at Tribhuvan University from Nepal with a core subject, General Microbiology, Biochemistry, Pharmaceutical Microbiology, Public Health Microbiology, a Medical Microbiology, and a research work. Then I studied Masters of Science in my Medical Biotechnology by coursework from 2020 to 2021 with the major subjects flow cytometry, microscopy and imaging, proteomics, and biotechnology solutions to infectious diseases. Then I transferred my coursework to research work, and I recently completed my honors in medical biotechnology in 2022 on the topic in vitro evaluation of luminescent nanoparticles for controlled release of drugs towards the treatment of type 1 diabetes. So during last year, final year of my undergraduate degree, we needed to carry out a project work, and I decided to work on a comparative analysis on the microflora of gastrointestinal tract in infants born by normal and cesarean birth method. This research is important. Like I chose this research because the delivery mode is an essential part of the development of the infant gut microbiome, and to investigate which birthing method is good for neonates, because neonates born by normal delivery have a microbiome like that of their mothers, and they have good microbiome in them. In contrast, neonates born by C-section, they lack good microbiome and are susceptible to diseases like asthma, diabetes, and many other diseases. So the research activities included in this project was culturing microorganisms, I did this by isolating microorganisms from stool of neonates and cultured them in different agar plate. Then I differentiated the microorganism species by doing different type of staining like gram-positive, gram-negative staining, capsule staining, spore staining, and performing various biochemical tests to differentiate the species, microorganism species. Finally, I performed antimicrobial susceptibility testing to test whether the isolated microorganisms are resistant to antibiotics or not. So the findings from our project is when birthing choice options are available, it is found better to consider normal delivery. Since a more normal colonization pattern of organism was found to be associated with this delivery mode, opportunistic pathogens were recovered from infants on formula diet. The study recommends breastfeeding for infants at least in their first few months of life. Finally, normal delivery should be given priority rather than C-section delivery so that infants develop non-pathogenic and mother's microbiome, which will help them shape their gut microbiome. My second experience on research is I recently completed my honors project on the title In Vitro Evaluation of Luminescent Nanoparticles for Controlled Release of Drugs Toward the Treatment for Type 1 Diabetes mellitus under the supervision of Dr. Helen, Dr. Lana MC Clements, and Professor Bernie Tup. So type 1 diabetes is an autoimmune disorder, meaning body's immune cells attacks its own cell, thinking it as a foreign invader. So it is mostly associated with children to adult, but anyone can get type 1 diabetes at any age. It accounts for all 5% of the diabetic cases, and the treatment is insulin therapy or islet cell transplantation. So the detailed work in my honors project was the fabrication of smart nanocarrier system with luminescent nanoparticle as a core and mesoporous silica as the cell, then co-loading of immunomodulation agent and a photosensitizer, then monitoring the drug release profile with and without near-infrared light irradiation, and also encapsulating the nanocarriers into sodium alginate capsules and characterizing the drug release from the capsules with and without near-infrared light irradiation. These two figures are from TEM. Here we can see that the upconversion nanoparticles are uniformly distributed in the mesoporous silica. So this is the preliminary data from my honors project. So in this, first I use insulin as a drug model to optimize drug loading and drug delivery before using NCC950, which is the immunomodulatory agent in this group, because NCC90 is expensive. And first of all, I loaded the drug and released, and I detected the insulin release with the help of UV-vis spectrophotometry. And in the graph, we can see that it shows the general release profile with sharp increase up to 7 hours, then decrease from 8 hours, which tells us that it is the accumulated drug release over time. In figure B is the release of MCC950. And since MCC950 is small molecule, which cannot be detected with UV-vis spectrophotometer, we detected the MCC950 release with the help of mass spectrometry. So we can see in the figure B that the graph shows the general release profile with sharp increase at first 0.5 hours, then a fluctuation over the rough plateau over the all period from 1 to 10, although there are some fluctuation. We can also see that the near infrared light had higher MCC release compared in MCC release rate compared to without near infrared light. Since I was only able to do MCC950 release with and without near infrared light just once because of COVID restrictions and lab lockdown, I need to further optimize this data. And second is my preliminary data from in vitro work from cells. I did cell proliferation assay. The cells were treated with different component of nanocarrier and for Min6 cells and fibroblast cells for different time points. And it can be seen that there was no toxic effect on fibroblast and Min6 cells with each component of nanocarrier. So from this study, the smart nanocarrier system was successfully fabricated and 2 drug molecules were successfully loaded into a core cell nanocarrier. We can use near-infrared light to control the drug release compared to the one without near-infrared light. The components of the nanomedicine system at relevant in vitro concentrations are not toxic towards Min6 murine islet cells. Mild toxicity was observed with MCC950 and photosensitizer on fibroblast cells at 48 hours treatment with higher concentrations, which demonstrated that the treatment effect from MCC950 to fibrosis. The preliminary data collected on the in vitro evaluation of luminescent nanoparticles for controlled release of drugs towards the treatment for type 1 diabetes mellitus demonstrated that near-infrared light controlled drug release was able to treat diabetes together with cell implantation. So I have written a review article on the title Challenges with Cell-Based Therapies for Type 1 Diabetes Mellitus, which has been submitted to the journal. I am also a co-author in a review article on multifunctional upconversion nanoparticles for photoacoustic imaging, which has been accepted in Exploration Journal. Finally, I am also a co-author in a research article, in vitro evaluation of the smart nanocarriers of core cell upconversion nanoparticles, mesoporous silica for drug release under near-infrared light irradiation, which has been drafted. So during my final year of undergraduate, I was on that time I was interested in research, which helped me to choose the masters of my medical biotechnology and ultimately helped me in choosing my honors topic. So I want to do a PhD topic on type 1 diabetes treatment using light triggered drug release. So why PhD? Since I have done my original research during my honors project, which has inspired me to take up the challenge for PhD. So I am interested in translating a research to clinically practical research with a powerful impact on society and contribute to healthcare, and also to become an expert in exploring solutions to type 1 diabetes and share my research finding with others. I find that a PhD can pave a new path for my long-term career goal. And why this research? Research for type 1 diabetes is necessary because when people are diagnosed with type 1 diabetes, they experience significant health concerns because of the disease itself has proven to be the catalyst for the other health problems like heart disease, kidney failure, high blood pressure, blindness, and nerve damage. People with type 1 diabetes or the parents of young children with disease must do the work of blood glucose level in body, monitor their blood glucose levels every day and night, and administer insulin through injections or an insulin pump, which is quite painful. Thus, it is imperative to pursue research to identify prevention strategies and improve treatments while striving for a cure. So therefore, we hypothesize that the near-infrared light-triggered smart nanomedicine system using core cell nanoparticles, that is upconversion nanoparticles, to deliver anti-fibrotic agent or immunomodulatory agent in a controlled manner can prevent fibrosis of the transplanted islet cells for treatment of type 1 diabetes. The goal of this research is the practical implications of a successful outcome with the study will be possible to cause release of IMA locally with subcutaneous cell graft by light external stimulation to control drug release for type 1 diabetes. And this project will systematically examine the effectiveness of a smart nanomedicine system to release IMA, thereby enhancing the function of beta cells seeded into a bioengineered device and implanted in subcutaneously in diabetic animals. So my research plan for PhD is divided into 3 years. In year 1, I will be creating a smart nanomedicine system containing an immunomodulating agent. Then I will be encapsulating nanocarriers into sodium alginate capsules and monitoring its release from the encapsulated capsule using near-infrared light and evaluating the safety of nanocarrier in vitro on fibroblast and murine islet cells. This will be continued from my honors project. And moreover, I will also be working more on compatibility of with different cells and more focused on gene expression. So the research skill obtained are the synthesis of conversion nanoparticles and silica, encapsulating nanocarriers into alginate capsule, drug loading and drug release using near-infrared light. The lab access would be chemistry lab, PC2 lab, and MAU lab, which I already have access to it. So in year 2, I will be loading the capsules into polymer scaffolds and monitoring its release from encapsulated capsule using near-infrared light, then evaluating the safety of nanocarrier in vitro on fibroblast and murine islet cells. The research skills required will be fabrication of alginate capsules, encapsulation of nanocarriers and beta cells into the alginate capsules, then transfer the capsules into the scaffold pocket. Lab access will be chemistry lab, PC2 lab, and MAU lab. In year 3, I will be accessing biomedical implant in animal model to diabetic model for drug release. The research skills would be how to handle animals and subcutaneous transplant of nanocarrier. Lab access would be on facility. So the significance of this study would be the smart nanomedicine system can enable the controlled release of IMA, hence facilitating the prolonged and effective treatment for type 1 diabetes. The application of smart nanomedicine system may be expanded to other chronic diseases like cancers. And finally, the incorporation of drugs that is inflammatory inhibitors and or antibiotics into smart nanomedicine system may prevent infection after surgery for longer duration. My future plan after PhD would be to boost up my career in research, firstly by doing postdoc, then accumulating industrial experience for research translation, and finally by exploring the solutions to type 1 diabetes. Thank you.
Thank you very much, Prescripti. I think your overall structure of your presentation is really good and can be like a model for other PhD candidates. Thank you very much. And is there any questions from the audience?
I have one question.
Scientific question. Oh, wait, do you want to?
Yes. When you talk about the gene expression in your presentation, what gene you want to investigate? I thought this is a drug. Or you want to do some signaling pathway or some?
Actually, due to COVID restrictions, I was not able to work more properly in my honors project. So I would like to, since we are doing a drug testing in the cells, so I would like to see gene expressions like how the protein is expressed, and also the different type of genes. Like I forgot about it, but I can show it if you want.
Yeah, that's fine. As long as you have the idea about gene, what kind of gene target you want to investigate.
Actually, I have done that, but I didn't get this, and like it's not, but I can show that if you want.
I guess,
Yeah,
you
yeah,
know,
yeah.
let me follow up that question, you know, because of Taiwan diabetes is affected by multiple genetics polymorphism. So the question is which polymorphism or which particular gene, you know, you want to evaluate in terms of gene expression. And I think that is a very important questions because you have to be clear, you know, what gene you want to do.
Yeah, so since this project is overall about the preventing fibrosis in the implanted device, so when we implant a device in subcutaneously, like the device works for at least 90 days, then after sometimes the fibrosis occurs on the scaffold. So our main goal is to prevent the fibrosis on the scaffold so that the device can work for a longer period of time. And for gene expression, like we are looking, like I'm looking into the genes, like for
I mean, you know, gene expression, you just simply, you know, look at, you know, which one's active, which one's not. Essentially, that's what it means. But because of your outcome, is you are talking about fibrosis. So it's not about the disease, you know.
yeah,
So that is something you have to make it clear, you know, in your hypothesis.
I think Tuan always get very good suggestions, especially for us like supervisors needs to consider.
Yeah, it's an endocrinology thing, so that's why, you know, I get used to it. You know?
Okay. So I actually have some question. So for the technology, how is it going to like compare with other like nanomedicine based technology for drug delivery? Like what's your innovation and the benefits here?
So I will be using near infrared light. So this is mild and there is no photo damaging effect from near infrared light. And another reason is it penetrates deeply somewhere around 30 mm to 1 centimeter compared to visible and UV light. And also it is safe and it helps in controlled delivery of drug.
Thanks. And for your like a 3 years plan, I didn't catch it very clearly. Like what's the difference between year 2 and year 1? How are you going to move on from year 1 to year 2?
So in year 1, I will be creating a smart nanomedicine system. Then I'll be incorporating that into alginate microcapsules. So in year 2, for that alginate microcapsules, I will be loading that into the scaffold. So basically, when we implant subcutaneously, we will be implanting the scaffold, which contains alginate microcapsules, capsules.
Okay, thank you. Is there any other question?
I have a question. It's a how can you prevent systemic delivery of your drug? So what makes sure that you only target the specific cell lines or specific cell types if you use in
So for cell lines, we will be using mean 6 cells and fibroblast cells. So for fibroblast cells, because
I meant when you use it as a subcutaneous device, what's the guarantee it will not reach other tissues or other cell types?
No, yeah, I did in honors project, I just did for mean 6 cells and fibroblast cells. But in my PhD, I need to work more on different types of cells, like different types of immunomodulatories and not only MCC 950 and other different types of cells.
Yeah, I think I probably can help a little bit here. I think your just question is how can you make sure the drug nanocarrier, when it release drug, the drug wouldn't go to other cell lines, but goes to targeted fibroblast cells.
Yeah, that was a question. So how you prevent that the drug enters another tissue which you don't want to drug?
But let's put this. Let me put this way, right? Where does the drugs metabolize for Taiwan diabetes? Do you know where does it metabolize? I presume in the liver or kidney or.
The drug is the
Where does it metabolize?
modulating agent. So it's mainly not to treat the diabetes, to treat the fibrosis.
All right.
Yeah.
But there have to be a place where it metabolizes. You know what I mean? For example, in our bone, it normally metabolizes in the liver. And that's why, when we study about metabolism of drugs, we focus on the liver cells.
Yeah, this drug is because we will do the subcutaneous implantation, so it will metabolize into the blood. And then that's one of the work we're going to do with the animal model. So we actually started the preliminary data with the animal model, and we collect some blood samples from the mice, and then we will do some analysis. And yes, it will metabolize into the blood.
Yeah, in blood, but I mean the organ that is actually metabolized. Different kind of drug. I can give you an example. Rosiglitazone, it's a very common drug, for Taiwan type 2 diabetes. But they normally metabolize in the liver.
Yeah.
And that's why, it has a major effect on the liver among these people, in these patients.
Sure. Yeah. I think we at this moment for these subcutaneous devices or implants, we use the immunomodulating agent, and we think the main kind of like metabolism stream is to the blood. We haven't collected other organ. We haven't done any organ pathology analysis. But the blood sample analysis is on the way. Yeah. Well, but definitely that's a good suggestion. We will check it up whether it will goes to organ, whether it goes to the liver, or whether it goes to the spleen or some other organs. Thank you for the suggestion.
Okay, I think if there's no more questions for these candidates here, let's all thank our candidates. You all did a very good job, and good luck with your PhD application. And especially thank Tuan and Majid and Yogi for coming here. And I think Wei already sent a team link for his candidates. Unfortunately, they can't join because they're overseas. So if you could please just go to the team link.
Yeah. I will send a link to the panel.
Okay.
So okay, thank you very much.
Thank you, Tuan. Thank you,
Okay,
Yogi,
bye.
Majid. Thank you.